In the virtual PCR lab, what you're going to be doing is setting up a virtual PCR reaction. What I'd like to do here is show you how we set up a PCR reaction in the lab itself. So what we have, first of all, are a set of patient samples that we have collected DNA from each of the family members where we'd like to analyse whether they have a mutation in the dystrophin gene. And what we're going to do is we're going to transfer those into these special PCR tubes that I have here. And these are small, thin-walled tubes, and they're thin-walled so that the temperature can be transferred readily. The temperature changes from the PCR machine can be readily transferred to the contents of the tube. So what we need to do first is transfer those patient DNA samples into those special PCR tubes. So here we have our first patient sample, and I'll use a pipette to collect that DNA sample. And then I'm going to transfer it, as I said, into this special PCR tube. And once I've transferred the DNA there, I remove my pipette tip to make sure I don't contaminate the other DNA samples. And then I put on a fresh tip. And what I need to add to that DNA sample is a buffer that contains all the salts that we need to maintain the correct pH for the functioning of the Taq DNA polymerase, the enzyme that's going to carry out this reaction for us. Also has magnesium ions because the DNA polymerase requires those to work. And it also has in there the DNTPs, the deoxyribonucleotide triphosphates, the raw material that the Taq DNA polymerase will use to build those new DNA strands. So I add that, get rid of the tip because I'm now going to add another reagent, and that is the dystrophin primers. The primers that are needed so that the Taq DNA polymerase will specifically amplify exon 50 of the dystrophin gene. So those primers anneal or base pair to the sequence just around that exon 50. And so when we add those, it's that region that the Taq DNA polymerase will amplify. So now I've added that. I again remove the tip. And so now what I'm going to finally add is the Taq DNA polymerase enzyme itself. And this is the enzyme that carries out that very special reaction. And remember, it's heat stable because we're going to have to heat a DNA sample up to high temperatures to get the DNA to denature, to separate the DNA strands. So it has to be a special heat stable DNA polymerase that we're using. Once I've added all the reagents to the first tube and I've gently mixed it, I then would add the patient DNA samples to the other tubes and all the reagents to all of them. Once it's been added to all of those, then what we're going to do is transfer the tubes to the PCR machine. And the PCR machine is a machine that will actually carry out all those cycles that we need to amplify the DNA polymerase. So it has a special heating block that I'll transfer the tubes into. And that's why they're thin-walled tubes, so that as the temperature changes very rapidly in the PCR machine, the thin-walled tubes allow the sample inside the tubes to also rapidly change in temperature. And so it will heat the samples up to about 95 degrees so that they'll denature, so the DNA strands will separate. We'll then cool it down to 55 degrees so that the primers can anneal or base pair with the sequence just around exon 50 of the dystrophin gene, so that when the temperature's heated up to 72 degrees, then that's the temperature at which the Taq DNA polymerase works best, and it will carry out the chain extension just at those sequences, and that will create large amounts of that DNA sequence only. So what I need to do now is actually set up the PCR reaction. So I do that and then I start the reaction. This will take a couple of hours. When it's finished, we'll have sufficient DNA sample that we can analyse by gel electrophoresis.